Assalamu alaikum students. Today I am gonna give you an overview of vitamin B12. You had this lecture yesterday with Dr. Asghar, and I'm just gonna repeat and highlight some important things of vitamin B12. So actually, first of all, we will be seeing the structure of vitamin B12. Now, this vitamin B12, this has a cobalt in the center, as you can see here. This is a cobalt, and then it has these pyrrole rings. Okay, this is a pyrrole ring. This 4 pyrrole rings. Okay, these pyrrole rings, when linked together, they make a ring that is Corin ring. Okay, so these rings and this Corin ring with cobalt, they are the main core of the structure of vitamin B12. Then there are some coordination bonds that are made here. If you see here, this is dimethylbenzimidazole. Okay, the lower one is attached with dimethylbenzimidazole, and this upper link is mainly with cyanide in commercial preparations. Okay, with cyanide, so known as cyanocobalamin. And then there are these physiologically active forms, which is deoxyadenosylcobalamin, if this deoxyadenosyl group is attached, and if methyl group is attached in at this top area, then it is known as methylcobalamin. Okay, so this 5,6-dimethylbenzimidazole is attached to the nitrogen of the cobalt. Okay, this cobalt has this nitrogen group, and this is attached with dimethylbenzimidazole. So you have different forms. Cyanocobalamin is a form which is used in commercial preparation. Then you have these deoxyadenosyl cobalamin. Then you have methylcobalamin, and thus these are the ones that are used in your biological processes. Okay, so what are the sources of vitamin B12? Vitamin B12 is mainly synthesized by the microorganisms in your gut. So mainly the main hub of this synthesis is your gut. Okay, and then there are some appreciable amounts of vitamin B12 in liver, in cheese, in if you see plant sources, you can get them in fortified cereals and soybeans, and then you can get them in red meat, eggs, poultry, liver, some crabs, crustaceans, cheese and all. So these are the sources of vitamin B12. Now, the transport of vitamin B12 is very important, okay, and it is usually really asked in the exams so many times. So what happens is that this you eat something, there is free B12. This free B12, it binds to a glycoprotein, which is an R protein. Okay, this R protein with B12, when acted upon by pancreatic enzymes, this R protein gets detached from the B12 protein. Okay, and then there is an intrinsic factor which is released from the parietal cells, which binds to the B12. Now this intrinsic factor B12 complex, it goes to the ileum. Okay, and here the mucosal cells have receptors for intrinsic factor. This intrinsic factor in B12 complex, it gets attached to the receptor. This B12 is sent into the mucosal cell and then sent into the blood, where this B12 binding proteins take this B12 to the liver to get stored, and this intrinsic factor is then it goes back. It doesn't enter the mucosal cell. So this is how your B12 gets absorbed in your body. Now, okay, now this is the absorption. We will be talking about the deficiency in regards to this absorption in later slides. Then we come to metabolic functions. So there are some reactions in which B12 is used as a coenzyme, and those reactions, one of the most important reaction is resynthesis of methionine from homocysteine. So homocysteine is converted into methionine using methionine synthase. Okay, here vitamin B12 and folic acid, the N5-methyl form of folic acid is used, and thus this methylation converts homocysteine to methionine. Here N5-methyl tetrahydrofolate, when methyl group is given to homocysteine, it converts into tetrahydrofolate, and homocysteine converts into methionine. So one concept of folate trap hypothesis also comes from this reaction. It is that in the lecture of folic acid, I told you this folate is converted into N5N10 methylene form. It is converted into N5-methyl form and all those things that can then synthesize purines and TMP. Those are the things that are necessary for DNA synthesis. Okay. Now what happens that if you have B12 deficiency, if there is a deficiency of B12, your N5-methyl form has come to work for conversion of homocysteine to methionine. But it cannot work alone. It needs B12 for the whole reaction to occur. But if there is no vitamin B12, N5-methyl form will start accumulating in the body. Now this N5-methyl form cannot be converted into the methylene form, okay, or the other forms. So this N5-methyl form is getting accumulated. The other forms are being used and they are decreasing. So your synthesis of TMP and purine synthesis is decreasing, ultimately leading to defective DNA synthesis, especially in bone marrow, erythropoietic bone marrow, and ultimately you're getting blast cells and megaloblastic anemia. Okay, so this N5-methyl form gets trapped, this folic acid gets trapped in the N5-methyl form in your body, and thus this hypothesis is known as folate trap hypothesis. We will be discussing this again in the later slide. ये मैंने आपको इसलिए बता दिया क्योंकि we were just discussing this reaction. So the second reaction in which vitamin B12 plays an important role, you know, many fatty acids and all, they break down into methylmalonyl CoA. Okay, now this methylmalonyl CoA using the enzyme mutase has to be converted into succinyl CoA, and this reaction needs vitamin B12 as a coenzyme. If there is deficiency of vitamin B12, your methylmalonyl CoA is not converted into succinyl CoA. Ultimately, these fatty acids, they get accumulated in your body, they get accumulated in the membrane, they get embedded in the membrane, and ultimately you get the neurological symptoms, neuropsychiatric symptoms that are associated with B12 deficiency. Okay, now there are other symptoms which are dizziness. You can get mood changes and these skin lesions. You can get fatigue, and the sensation of pins and needles is like some neuropathic things, and along with the neuropsychiatric symptoms that I already discussed. Okay, then what is pernicious anemia? Deficiency of vitamin B12 is causing pernicious anemia. So pernicious anemia, you know, intrinsic factor is needed for the absorption of vitamin B12. If intrinsic factor is not, that B12 cannot be absorbed. Now, if there is deficiency of vitamin B12 or there is malabsorption of any kind, what will happen? You will not have intrinsic factor. So if there is no intrinsic factor, this deficiency will, there will be no vitamin B12, there will be no vitamin B12. Ultimately, you cannot, there will be no vitamin B12, and this will lead to anemia. I already just discussed the megaloblastic anemia. So any anemia which is due to intrinsic factor deficiency, is known as pernicious anemia. You know, the same sort of anemia, megaloblastic anemia, occurs in folate deficiency also. So there you have to recheck that whether the megaloblastic anemia is due to pernicious anemia or vitamin B12, due to vitamin B12 deficiency or due to folate deficiency. And for that we do a test which is a Schilling test. I'll be telling you what the Schilling test is. So this is pernicious anemia is a chronic illness by impaired absorption of vitamin B12 because of lack of intrinsic factor deficiency. So Schilling test, basically we don't need to know it in detail, but Schilling test is a test where you try to find out the reason of vitamin B12 malabsorption, okay, whether it is dietary deficiency, whether it is intrinsic factor deficiency, whether it is some bacterial overgrowth, whether it is some pancreatic insufficiency that is causing this vitamin B12 deficiency in the body. Okay, so here they use radioactive B12, giving it intramuscularly and orally, and then checking the 24-hour urine B12, radio labeled B12. So by that test you can find out that if supposedly you've given B12 and the whole B12 is coming out in the urine, that means the B12 is not being absorbed. But if you give intrinsic factor along with B12 and there is less low B12 in the urine, that means now you had given intrinsic factor, B12 is being absorbed. That means this person had intrinsic factor deficiency. That's why the B12 was not being absorbed. So this is a Schilling test which is necessary, or which is used to find out whether the deficiency is due to whether the deficiency is due to intrinsic factor deficiency or some other cause. Okay, so I think this is actually a folate trap. I have already discussed with you that folic acid is being trapped because of B12 deficiency, and this B12 deficiency actually is occurring due to any reason. There's a B12 deficiency, N5-methyl is trapped in that form and it is not being converted into any other form, and this can lead to accumulation of the N5-methyl form and the decrease in the other form, because they are already doing their function. So they are not, this form is not being converted into that form. So this is the form that is higher in your body and it is trapped in your body. So this is folate trap hypothesis. So what you can do is the hematological abnormalities of B12 deficiency sometimes partially can be reversed by folic acid, and to prevent the CNS effects of B12 deficiency, when you get megaloblastic anemia, you start B12 and you start folic acid also. Once the reason is found out that it is because of B12 or folic acid, then you can stop the other vitamin. Okay, so this was already discussed in the folic acid lecture, how folate is converted into dihydrofolic acid and then tetrahydrofolic acid, and how then there is this methyl and methylene form that can form TMP and DNA synthesis. Okay, so Sir Asghar had already discussed this. I was just giving you an overview. Hope you understood what was being taught, and that's all. Thank you very much.